So, we are going to molecular docking. What is molecular docking? Actually, molecular docking, so one second, this is the last topic of today. We will use Swissdock is a software which from Expressive only we are using it for the molecular docking purpose. So, molecular docking. Molecular recognition. What is molecular recognition? Molecular recognition is the ability of biomolecules to recognize other biomolecules. So, the ability of the biomolecules to recognize other biomolecules are called molecular recognition, and selectively interact with them in order to promote the fundamental biological processes such as transcription, translation. Lot of molecules are interacting each other, transcription factors, RNA polymerase, different proteins interact with each other and they selectively interact. So, RNA polymerase has different subunits, they selectively interact with each other and help in various fundamental processes of our body: transduction, transport, regulation, enzymatic catalysis, viral bacterial infection, immune response. All these proteins are interacting each other. Molecular docking. Molecular docking is the process involves placing molecule in appropriate configuration to interact with a receptor. So, molecular docking is a natural process that occurs within seconds in a cell. Usually, molecular docking process is occurring in a cell every time, means molecules have been interacting each other. Okay, molecules has been interacting with the receptors, hormones have been interacting with the receptors, and proteins have been interacting with the receptors, small molecules have been interacting with the receptors. So, this is natural process which occurring in the every cell. In molecular modeling, the term molecular docking, when we go for a bioinformatics, the term molecular modeling refers to study of two or more molecular structures fit together. How we can fit two or more structures together? How we can study the interaction with the two or more structures? That process is called molecular docking. Usually, the molecular docking mainly interaction will be between two molecules. One will be a, is there? Yes, it will be there. So, mostly the in docking the interaction will be between two molecule. One will be a receptor and other will be a ligand. So, the interaction of the receptor and ligand can be used in the drug designing process, because the receptor are the ones which are mostly any receptor can be enzymes, proteins. So, receptors are the binding of molecules to the receptors can bring about drastic changes in the cell. A lot of the signal transduction pathways occur which can result in changes in the cell. So, in molecular docking in silico condition, we are looking at the interaction between the receptor and the ligand. So, computational docking discipline we are going to study. So, difficulties in obtaining experimentally determined structural data of macromolecular complexes triggered the development of computational predictive methods. We have predicted the structure of a protein now only. The computational docking. So, in order to do the try a lot of drugs to the animal model or in vitro cell model, it will take a lot of time. So, if we can model that interactions between a ligand and a receptor using in silico techniques, that process is called computational docking or in silico molecular docking or just docking. Is a computational science aiming for predicting, everything is predicting in bioinformatics, nothing is experimental, everything is predicting. The predicting the optimal binding and orientation and conformation of interacting molecule in space and estimate the stability of their complex. So, that is what we are checking by molecular docking. We are checking how this, using what type of bonding or how these two molecules, the receptor and ligand, is interacting, in which orientation or in which conformation, how they are interacting in the space, in the 3D space, how much stable is their interaction. So, all these will be determined, all these will be are the main factors which is involved in molecular docking. I am not going to drug designing or anything. There are several qualities of a drug, all these things are there. We are just studying the molecular docking only. Drug designing, we need to think about various drug parameters, all these things. I am just dealing with the molecular docking only. So, the prediction of how a ligand molecule bind to the receptor, what is its orientation, what is the conformation of the interacting molecules in space, and they estimate the stability of the complex, how much stable is the complex between the ligand and the receptor. So, molecular docking predicts whether or not two molecules interact. Molecular docking help us to find out whether these two molecules will interact or not, how much affinity they bind, and the 3D structure of the complex. So, all these things can be assessed by molecular docking method. So, computational docking is essential component for the modern drug discovery. So, we need to study more about drug while choosing the ligand. So, it is not such easy thing. So, we need to think about the various ADME properties of drug, all these things, lot of things we need to study before choosing is a drug. I am just now blindly selecting one drug and trying to dock. So, it is not such simple. I am just showing what is docking, that is all. Over last few decades, it is routinely and successfully applied to most pharmaceutical and biotech companies in large number of applications. So, it is docking makes speed up the production of pharmaceutical drugs, as well as biotech companies are using this docking as the initial tool for designing the drugs. So, molecular docking classifies biomolecules. Mainly docking can be of different types. One is a protein ligand docking, protein and its ligand, protein docking, and nucleic acid protein docking. We will be dealing with protein ligand docking only. So, interactions between small molecule and the protein, that is what we are going to study, protein and ligand. Ligand will be a small molecule and protein, it will interact with the, is far much better understood than those between the protein and nucleic acid. So, mostly studied is the protein ligand docking, and that is what we are going to perform. So, the result part we will see after the result. So, all this for molecular docking, you can for the docking files you can store in a separate folder. I am not storing in a separate folder, anyway I will show you. So, Swissdock is the software which we use, not Swissmodel. So, Swissdock is the software we use. It is an online docking software. You can see this page. You can go to the submit docking here, where you can submit your target and ligand and your project name and email, and you will get the result in the email. And where we will get the target? We need to go for the PDB. Okay, protein data bank only we will get the target. Some link is there. So, I will go to protein data bank. Okay. From protein data bank we will get the 3D structure. If you have the 3D structure, you can use that. Your experimentally determined 3D structure. I am just searching a small protein. I have just run a protein prion only. Run a protein earlier, but it was not working because my memory is less. So, I am again selecting a prion. Usually prion protein structure is more small. So, I am just downloading it PDB format. Okay, PDB format. Okay, usually this okay. Then where we get the small molecule ligand? For getting small molecule, we have a website called PubChem in the NCBI. Just type PubChem. you will reach there. It's a website which contains small chemical molecules which can be used as a drug. So, you can get a lot of molecules here. Aspirin, I will select aspirin for docking. You can just select aspirin. So, aspirin structure we will get here. You can click aspirin details, all these things. I think you know how the PDB file is done. I am in a bit speed, but I have shown you how to download the PDB file. You can click the aspirin so that every details of aspirin is there, but every details I do not have time to read. I am just downloading the aspirin and we are going to dock the prion protein with the aspirin, just to show you only, because if it is this big protein, it will take a lot of time. Just click the download and we need to store it in the SDF format. Okay, save it in the SDF format. There are a 2D confirmation is there, download data to display in the page is there, and the 3D confirmation SDF file we need to download. Here you can see download, click here, and the 3D confirmation, we can save that SDF file. So, if we just we have the ligand file and we have the the PDB file of the model. If you just upload that file in the target, that protein file and ligand file, they will not accept, this software will not accept. Usually the protein file mostly it will contain some ligands or water molecule in that. So, this prion protein file may contain any other protein. When you do, you can, if you have a very good system with memory, you can use that system. We need to go for a preparation of the protein as well as the ligand. So, simple preparation we are doing. We need to remove any ligands present and we need to remove any water molecule present in the structure. If that water molecules are present in the structure or ligands are present in the structure, this Swissdock will not accept the structure. So, we are just removing using Discovery Studio. We have seen Discovery Studio where we have visualized the structure of a spike protein. We are just opening our prion protein structure here. Open. You can just download Discovery Studio Visualizer, where you can do the preparation for the protein as well as the ligand. So, after doing the preparation only, you can upload in the Swissdock. For that, you download a Discovery Studio. It is a very free software. You can open that prion protein, where it is, I think it is in my downloads. Yes, where it is. No. I think this is the one. This is the no. I will check. Here is the prion protein. I think file is IAG2. Okay, IAG2. So, I am just taking this IAG2. Okay, and open in this. So, these prion proteins, if you take other proteins, you can see water molecules, ligands. I think this is a simple protein. I think it will not have any ligands or any water molecule. What you need to remove, for removing the water molecule or ligands, what to do is that I will show the earlier molecule. I will show that, but docking I can't use that. What earlier molecule we have, say, you can think. Yes, this molecule. Okay, this molecule I will show for this molecule. So, go to this scripts. Okay, scripts, and we can select the water molecule. Select the water molecule. It's a big structure. I don't know whether they come. No water molecule found. Just so it will be shown in yellow color. At least I can see some ligand. You can select ligands. Yes, you can see ligands in yellow color. You can see ligands in yellow color. Like that, water molecule will be also be shown in yellow color. You can see ligands, okay? Extra ligands, metal ions, all these unnecessary molecules. It is necessary for the function of that protein, but it is not associated with that protein. So these ligands, you just select it, the script select this, and you click your delete button in your computer. Just click your delete button. So ligands will be deleted. This is one of the, and water molecule also same thing, select it and delete it. Okay, that is what we need to do for the preparation of the this target protein. So target protein, water molecules and ligands should be removed. So it will be, when we see, take the, we can see in this protein, our protein scripts, selection, water molecules, there is no water molecules are there here, small protein, and scripts, if water molecule, script, select water molecule and then delete that. Okay, water molecule will be shown yellow and we can delete that. And again scripts, selection, ligands, there is no ligand also, small protein, for the fastness in the docking only I have selected this protein. So there is no ligand. If it is ligand also, you can click your delete, so that the ligand will be deleted. So then just save this protein after the preparation of the target, save this protein without any space in the file name. We can just add a P1 I think, just P1 here, so that it can, and it should be saved in the PDB format. You can see the protein data bank format here. It should not be stored in the Discovery Studio format, PDB format. Okay, we have prepared the target protein, and ligand preparation can be also be done here. You can open the ligand here. Our ligand is aspirin. We can open the ligand, and the ligand preparation is just saving it in a mol2 format. We can open the ligand here and we can save it in another format. It is in the SYBL mol2 format. This format is accessible with the Swiss model. So mol2 format we can just save it. So the ligand preparation and the target preparation is now over. Our ligand and target is there in the our file. You can make a separate folder also, and when we are uploading, we can see sometimes they will not accept that. So we can, we can click the upload file here and we can upload it. Sometimes they will not accept. You can just upload that prepared target protein. They will check that. So if our protein is not, you will give a tick sign. If our protein is not prepared by deleting the ligands, water molecule, without deleting the ligand, water molecule, they will not accept. So we can upload the ligand also, same way. Ligand also we have prepared in the mol2 format. We can upload. Let us see whether they will accept or not. Yes, accepted. We can give the job. I can see project 1. Okay. Project, I think only alphanumeric characters allowed. So it is required one project. I just give email address. You can give. It will be given to your. It's optional only. You can start the docking. So docking, the result will be available here. It's very small small molecule, so we will get the result, hope so. And the result page, you will see a table which contain the delta G value, which indicates the Gibbs free energy, and that we can see the ligand will be docked to different areas of the particular target molecule, and we can download the data and find out which area binding which is needed for you, whether the ligand is binding to the active site is the one you are targeting. You can check which area of the protein it is binding, and and from the delta G value you can you can find out that how much firmly or how much effectively the ligand is binding to the target. So, all these things we can see from the result, and that is all for the that is all for the docking. We will wait for the result. Okay. So, just wait for the result. This is all about our workshop. Oh, not yet terminated. Okay. Sometimes error, all these things will be coming. So, we can wait for. So, let us see the result. This is so actually in the result we will have we will just read the result. Usually it will have a table. How we can show the table. So actually where is the result? No result. I will just show you the table. Doc result. We can see maybe we can see the image. I hope we will get the image. Yes, we will see the result like this table. We can see the table like this. I will copy that and paste it in a. Let the result come, we will see the result and go. Anyway, I will copy that picture here, because if this type of condition can, I thought the homology modeling will also not work. So, this type of server error can occur. Actually, you can see what is in the table. Usually, they will give the structure with the ligand, and we can see what it is present in this table. Actually, I will paste the table here. This is after downloading they have shown. We are not doing that. So, like this table will come like this. Usually, it has it's a table, and each the ligand has been docked this number of times with the different residues of that structure. So, you can select which docking we want, and depending on the delta G value. The lower the delta G value, the better the docking, and how it is fit, that it is determined by the full fitness. I will show the definition of full fitness. Full fitness determine how effectively the ligand fit to that of the structure, as well as the delta G, the Gibbs free energy, will tell you that lower the Gibbs free energy, these are very low values, 13 point. Lower the Gibbs free energy, the better the binding within the ligand and the target. So, we can select, we can download this data, and we can view it in UCSC, not UCSC, some viewer is there, camera also we can see. So, in that we can visualize that and find out which binding is perfect, which ligand is, these are all the possibilities of a ligand to bind in the various region of the target. So, different possibilities they are giving. These possibilities are the best possibility. So, because they are giving the less delta G value. Less delta G corresponds to the most effective interaction between the ligand and the target. And if you need the ligand to be interacted with a particular position, you can download the data and check whether it is the ligand is interacting and we are getting a somewhat good delta G, you can select that region and go with that result. So, like that we can see, and I think we got the result. No. So, this is how we can see the result. I will again click here. I do not know whether result came. Yes, result came, yes, our result came. So, we can see the result of great, otherwise I would have stopped this workshop without any result. So, you can see different our delta G is not much lower compared to that of the result which we seen in the screenshot, but we can see if you click here, okay, and you can see as the this prion protein is so small, we can see very clearly how our aspirin is interacting with the prion protein. You can see, and you can download the data here, and you can use UCCSF camera, or else you can see it in different visualization tool and study how it is interacting. You can click here and see whether the where the ligand is interacting, to which molecule the ligand is interacting, and how the interaction is about, how strongly the interaction is about. Here the delta G is -6. So, so these are the first values are the best values, and the interactions you can check your interactions and finalize which interactions is suitable for your study. Depending on your study, when you are looking the ligand to be interact with a specific residues, you can specify, you can check that interaction is being having a good delta G, and thereby you can confirm that your docking is proper. So, usually Swiss model docking, people are publishing lot of research papers with Swiss model docking itself. So, this tool is very useful and easy for docking. It is not it is very useful tool, we can say. So, you can see in the table you will get show the first column of the table is I am just winding up, don't go away. First is show. If we click that, it will show where the ligand is binding. Okay. Just I am interpreting the so that I can. It's a radio button. By clicking that any one of the radio button, it will display the confirmation of the complex against it will present the ligand, how the ligand interact with the target. We can see if you click each show. Okay, these are the this table list. What the table list? Table list the interaction of the ligand with the various parts of the protein, target protein. Okay, you can see the ligand is moving. Is it okay? Okay. And these delta G indicates how much tightly or how much stably they are interacting each other. So, next two columns provide the number of confirmation elements column and against each cluster number. So, the number of confirmation and cluster number will be giving in the next two columns. So, number of confirmation and cluster number will be giving in the next column, and so number of confirmation in the element column and the cluster number will be given the cluster column. Then the next column will provide the full fitness. Full fitness is a bit confusing thing. I will just read the definition. It's a bit difficult to understand. Then next column will be the estimated delta G, that is the Gibbs free energy. That lower the delta G, the better the complex is about. And when you over okay, that's all. When you hover over the structural complex, you can see the what region you are. When we hover over means this. When we go through the structure, we can see which amino acid we are seeing, valine. Where we just go through arginine. So you can see where it is interacting. The histidine, I think HSD, amino acid, there is a 309. So, to this amino acids only they are interacting, the ligand is interacting in this conformation. So, last I think anyone is there, I do not know. So, delta G is reported by the docking software, in fact just scoring function which give very rough ideas about how preferable certain docking position is, about how the delta G give a rough idea that how the particular position, docking position fit, or how the docking particular docking position interact with the or the complex, how the particular docking position interact perfectly each other with between the complex and the ligand. It's almost never equal to the experimental binding energy. So, the delta G here shown cannot be similar to the anyway any experiments with bioinformatics will not be highly replicable to the experimental. So, the delta G actual delta G at that region, interaction between the ligand and the protein molecule will not be same as that when we do it in the simulated during bioinformatics tool. Only some loose correlation can get, we will get an idea whether that particular position can particular ligand can interact at the particular position of that target molecule. That is the delta G. Delta G should be small, that will be lower the delta G, the better the binding or the better the stability between the that position stability between the binding of the ligand to the template at that position. Okay. Several positions they are showing in the particular graph. So, at that position how the ligand interact with the template, the target. So, last point, full fitness. What is full fitness? Full fitness of a cluster is calculated by averaging 30% most favorable effective energies of its element. Full fitness means it is calculated by averaging 30% most favorable effective energy of its element in order to limit the risk of the few complexes penalizing the whole cluster. Effective energy is written as the sum of the total energy of the system and a solvation term. So, this is the full fitness is calculated by averaging 30% of the most favorable effective energy of its elements. So, that is the full fitness value. So, this is all about docking